Dear colleagues, as we unfortunately had to cancel this year's World Congress in Montreal, we thought it would be a good idea to at least share some highlights from the virtual Congress that took place in February. Before the video starts, I would like to announce that the 2022 World Congress in Taipei, Taiwan will take place from June 9th to the 12th. I look forward to seeing you there in person, and now please enjoy the video. Oh, and don't forget to subscribe to our YouTube channel below for free so you can watch more videos in the next weeks. Thank you.
Thank you for coming to our presentation. What I'm going to be talking about are some animal models that we've used in order to try to understand risk factors in schizophrenia, and particularly how stress impacts the system. To do this, I'll be using an animal model based on disruption of neurodevelopment using MAM, or methylase oxymethyl acetate. This is a DNA alkylating agent that we give during gestational day 17, which is equivalent to the second trimester in humans, which is known to be a critical period for schizophrenia risk. The MAM model recapitulates a number of disruptions seen in the human schizophrenia patient, including anatomical, such as cortical thinning and increased cell packing density, parvalbumin interneuron loss, behavioral dysfunctions, such as executive function, sensory gating, and affect, and pharmacological changes, such as increased response to amphetamine and PCP, and a hyperdopaminergic phenotype. There's substantial evidence for an involvement of the dopamine system in schizophrenia. However, there's little evidence for a primary deficit in the dopamine system. Instead, we now believe the dopamine system is probably normal, but is overdriven by other structures such as the hippocampus, which studies have shown is hyperactive in schizophrenia patients. Is the hippocampus hyperactive in the MAM animal, as it's been shown in imaging studies with schizophrenia? What we find is that the hippocampus is firing significantly faster. Moreover, it's also dysrhythmic in that there's a dropout of low-frequency gating rhythms and an increase in higher frequency rhythms. And this hippocampal hyperactivity caused children at risk for schizophrenia based on family history. The children that were hyper-responsive to stress tended to be the ones that converted to schizophrenia later in life. And stress is known to cause a loss of the parvalbumin interneurons, which are the same neurons that are lost in schizophrenia patients in the MAM model. Are MAM-treated rats more sensitive to stressors in this premorbid state, as is proposed to occur in schizophrenia? While looking at three different measures of anxiety: elevated plus maze, where they tend to hide in the closed arms, which is a measure of anxiety; increased foot shock-induced vocalizations, which shows increased response to stress; and increased firing in the basolateral amygdala, an area that's involved in anxiety and stress responses. So all three of these parameters show that in the premorbid state, the animals pre-pubertally are still hyper-responsive to stress and hyper-anxious. Now, if stress plays a role in the development of schizophrenia, what if we can circumvent the stress early in life? Well, we did this by using the anti-anxiety agent diazepam, and this was given at a dose that normalized activity in the elevated plus maze and brought amygdala neuron firing back to normal. What we found is if we gave this pre-pubertally, just for 10 days, withdrew them from the diazepam and looked at the MAM or control animals as adults, what we find is a blunting in the loss of parvalbumin interneurons, a failure to cause the activation of the dopaminergic neurons, so elimination of the hyperdopaminergic state, and a normalization of response to amphetamine. Now, this is using diazepam. Obviously, we don't want to give diazepam to children, most of which aren't going to convert. But what about non-pharmacological interventions? Well, to do this, we used environmental enrichment, where we put half the rats into an enriched environment with extra exercise wheels, extra toys, extra compatriots. And what we found is raising them between postnatal day 21 to 40 in the enriched environment prevented the increase in dopaminergic neuron firing and also prevented the loss of the parvalbumin interneurons. So just like diazepam, environmental enrichment seems to prevent the expression of this pathophysiology. So converging evidence from clinical and basic science studies suggests the inability to regulate stress early in life can lead to pathological changes in the hippocampus that may underlie the emergence of schizophrenia in the adult, and the number of dopaminergic neurons firing. This is more consistent with depression than with schizophrenia. Now, why are prepubertal rats more sensitive to the effects of stress? Well, during this prepubertal period, we're in a critical period. It's a period of enhanced plasticity where synapses are being made and broken and rearranged. After the critical period, the interneurons develop paraneuronal nets that protect them from further damage and also lock in the plasticity. However, we can reopen the paraneuronal nets by certain types of interventions, including HDAC or histodeacetylase inhibitors. So HDAC inhibition can reopen the critical period. Can we make adult rats vulnerable by reopening the critical period? So what we did is to give valproic acid starting at adult postnatal day 60. And then what we did is to start giving the combined stress at postnatal day 65 to 75. And then did our measures a couple weeks later. And what we found is that in the naive animal, the stress caused a decrease in dopaminergic neuron firing. But with valproic acid, the same stress now caused a more schizophrenia-like increased number of dopaminergic neurons firing, time when the parvalbumin neurons are protected by the paraneuronal nets. Now you tend to get stress-induced depression. So again, the common variable is stress and susceptibility to stress, but the timing of the stress determines the pathophysiology. So the increased response to stress, we think, may be mediated by a failure of systems that limit the impact of stress exposure. And one that we've studied a lot is the medial prefrontal cortex. Lesioning the medial prefrontal cortex is enough to cause a prepubertal MAM-like increase in responsivity to stressors and leading to a stress-induced schizophrenia-like state. So if the medial prefrontal cortex isn't functioning, you're going to have more susceptibility to stress, which is going to lead to these stress-induced damages. If you're protected during adolescence, you get depression. If you're not protected, you're more likely to get schizophrenia. If we can identify susceptible individuals based, for example, on early life tests for stress responsivity, and if we can intervene early enough, we might be able to prevent the deleterious effects of stress and the transition to schizophrenia and potentially alleviate the long-term consequences in adult affective disorders and schizophrenia. So again, this is consistent with what was shown by Eve Johnstone. It's also consistent with studies that are being done in Australia where individuals that are at risk are put in protective environments to try to mitigate the effects of early interventions. Now, to thank the people who did this work, especially Holly Moore, who developed and brought the MAM model to my lab, Yi-Zhuan Du, who did the work looking at some of the prevention effects, Stan Floresco, Tony West, who did the work looking at the circuitry of schizophrenia, Millie, Felipe, and Jason, who did the work on the neurodevelopmental model, Eric Zimmerman, who did a lot of the work looking at the vocalizations, and our collaborators. And I'd like to thank you very much for your attention.
Welcome, everyone. I'm pleased to contribute to our symposium on gene-environment interactions by talking about a convergence circuit for genetic and environmental risk for schizophrenia. The systems level question that we're asking here is trying to parse causal risk factors for schizophrenia into its component parts. For every illness, you can distinguish genetic factors, environmental factors, and their interaction. Of course, many genes are known, many environmental factors are known, and many are discussed in this symposium. What we're specifically going to do here is to ask the question, how do these genes, how do these environmental risk factors work? In other words, we're asking what neural systems are different in the brain of someone who carries a genetic load, say, who has been exposed to an environmental risk and resilience factors. And I do this by way of examples, but I'm trying to develop a more general point, namely that these individual risk and resilience factors converge on a common circuit. I'm going to lead in with an example that we've worked on a lot, and that is the by now very well established fact that has been known since the 1930s of the preceding century, that if you are born in a big city or grow up in a big city, your risk for mental illness is considerably increased. This is a famous map from a study of Chicago, Ferris and Dunham. Similar data have been found around the world. This is the first replication in Europe, which happens to be in my city in Mannheim, where we now currently work. And wherever you look, you find that if you're born in a big city, your risk for schizophrenia goes up by around threefold. And that is one of the most significant risk increases that we know overall in the risk resilience landscape. Now, many people have theorized about this, and the suspicion has been that that might have to do with social stress, with the way people interact with cities. So, now, we're now past, as a field, the state where we're looking at single genes. What we now like to do is to look at polygenic risk score, in which we add up all the common, at least, risk factors for a specific illness. When you do that and look at that then continuous polygenic risk score here for a range of brain imaging tasks that we've used, we found that yes, indeed, you can also here find that, for example, the cingulate cortex again exceeds the significant threshold test-wide for the number of tests we were doing here. So we have some indication that also polygenic risks impact on this. But on the other hand, we're also interested, of course, in epigenetics. So we want to move beyond genetic risk. To accomplish this, we have recently been able to establish, using a joint machine learning approach, to derive a reproducible epigenetic signature for schizophrenia from blood. So what we did here is we used relatively large samples to first, in the discovery sample, learn brain methylation based on blood methylation. As you know, methylation is one of the most important primary epigenetic molecular mechanisms. We validated that in whole blood in a case control sample, and we were then able to ask the question, these predicted brain methylation scores, are they linked to methylation in brain? Because that's what we want to know. And we did that in post-mortem brain. And are they related to brain function in vivo, which we measure using fMRIs? I'm going to show you a current example that just appeared in Science Advances, which concerns movement. You probably know that schizophrenia treatment through aerobic exercise has been a thing that people have been very interested in studying. So people on a treadmill, for example, but most of the exercise that we're doing each day is non-exercise activity. It means me going around my day, going from A to B. And this is something that we can measure in this experimental setup, both by GPS and also by these tiny sensors, which are in smartphones and also can be worn separately in fitness bands called accelerometers. And when we did this, we found that there was in fact a positive relationship between the amount of movement, non-exercise activity that you have and your well-being. And that has been related in our data here for the first time to brain, and again to the cingulate cortex. Interestingly, in this part, to the subgenuine cingulate cortex, which is a brain area that is especially important for regulating effective well-being, the very same measure that we found non-exercise activity related to. This is my final point. We can use this information not just for understanding risk and resilience in general, but also to target therapies, because we know that our current therapies are far or too far removed for precision treatment from the underlying biology, which is symbolized by the different colors of these people in here who are mixed if they come into our hospital and are insufficiently grouped by diagnostic categories. But if we understand the genetic, behavioral, and also topic of today, the neurosystems level concomitant. The hope is that we can then sort them into more homogeneous endophenotypes that we can then link to the necessary psychotherapies, pharmacotherapies, and preventions. In order to do this, you will need both highly multivariate data sets. And I've shown you today how data from genetics and epigenetics needs to be combined with imaging to understand these risk and resilience circuits, and you need the necessary computational firepower, especially approaches from artificial intelligence and machine learning are very helpful to do this. In this way, what I've shown you today isn't just basic science, it is that too, but it also has through computational neuroscience strong potential for leading to a precision psychiatry approach.